Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. We will now go to lecture number 45, which is the menthol story. So, where I will cover biosynthesis and pathway manipulation for enhancing the yield of menthol. So, the concepts covered first will be the biosynthetic pathway of menthol and its regulation and its cellular localization, and next we will discuss a few case studies where attempts were made to enhance the menthol yield in peppermint plant through genetic engineering approaches. So, menthol is basically a monoterpene, which I have mentioned in one of the previous classes. So, the structure of menthol, just to remind you, this is menthol, and another compound monoterpene which is not menthol, but it is called carvone. This is carvone and that one is menthol. Well, okay. So, this pathway originates from MEP. So, MEP means it is basically operating in the plastid, but then the substrate is basically the GPP, geranyl pyrophosphate, and then GPP makes limonene, and limonene subsequently converted into either carvone or menthol. So, the major difference is here that one enzyme which basically drives the limonene towards the carvone, which is limonene 6 hydroxylase. Limonene 6 hydroxylase, whereas the other enzyme which drives the pathway towards menthol, which is limonene 3 hydroxylase. Carvone you will find in the species which is Mentha spicata, and menthol you will find in the peppermint, which is Mentha piperata. Mentha spicata is also called this common name as pudina. So, now let us see this in detail in the next slide. So, that means the biosynthesis now we will draw. So, here at the cellular level, so multiple cellular organelles are involved. So, I will put it in this way. First, I will try to draw the organelle and then I will put the pathway inside. So, consider this whole thing as a cell, So, I am only putting here the green indicates basically it is the chloroplast. Okay. And then I will also put endoplasmic reticulum, which should be here. And then another endoplasmic reticulum, I have to put it here. And then I also have to put the mitochondria here. Okay, so this endoplasmic reticulum I have to make it slightly larger in size. Oh, sorry. Okay. So, now let us write. This is plastid. Okay. And this is endoplasmic reticulum, this is endoplasmic reticulum, and then this is the mitochondria. So, the MAP pathway starts with pyruvate and D gap, and so we will straight away start with the DXS, then DXS, and then it makes DXP, and which MAP, and then subsequently I will push the pathway in this direction. Finally, at one point we will see the formation of GPP. So, now I will write the compound. So, this is first one is and this is MEP, sorry, this is MEP, and the enzymes are and then so many finally we will land in GPP. So, the GPP is here. So, GPP will be converted into limonene. LIMONENE. This limonene will be transported out and enters into the endoplasmic reticulum. So, limonene I have to write it again, limonene, and the enzyme responsible for this step is limonene synthase. So, limonene synthase basically converts GPP to limonene, and limonene now moves inside the endoplasmic reticulum, and where by the action of this enzyme L3H, which is the 3-hydroxylase, limonene 3-hydroxylase, which converts limonene to, so I put L3H, yeah, which converts limonene to trans isopiperetinol. So, trans isopiperetinol. So, this trans isopiperetinol will be subsequently converted into isopiperetinone. So, the trans iperetinone will now moved into the mitochondria, and where it is converted into isopiperetinone. So, this is trans isopiperetinol now converted into isopiperetinone. Okay. This isopiperetinone will come out from the mitochondria into the cytosol, and then, so I have to put the writing again, isopiperetinone, and which will be converted into isopulegon, cis isopulegon. So, cis isopulegon, and cis isopulegon subsequently converted into pulegon. So, I put the arrow, and then this pulegon will move in one route. Pulegon will move inside the endoplasmic reticulum again, and where it will be converted into menthofuran. Furan. Okay. Whereas, in another case, this pulegon will be converted into menthone. So, pulegon can be converted into either menthone or also isomenthone. So, menthone or isomenthone. Okay. And from menthone, menthol is finally synthesized. And from isomenthol can be formed, and also neo-isomenthol can also be formed. isomenthol can be formed. New isomenthol can also be formed. So, we put the arrow here. Okay. So, this step basically occurring in the cytosol. Okay. Now, it is also possible that MVA pathway contributes towards the menthol biosynthesis, but much later it was found that in fact MVA pathway is not contributing to menthol biosynthesis, because there are some blockage exist in the late steps of MVA pathway. Therefore, I am not showing the MVA pathway here. So, as you know, the MVA pathway operates in the cytosol. So, you can keep a space for this. So, this is basically the MEP pathway, and here is the space for MVA pathway, which is not really contributing towards the menthol biosynthesis. Now, this is about okay. Now, there are few enzymes I have to put it here. For example, this important enzyme, the MFS, menthofuran synthase. So, MFS will be here, menthofuran synthase, and IPR will be here, which converts isopiperatone to isopulegon. So, and another important enzyme is IPGI. Okay, so I make it simple as I can. So, IPGI, yes, cis isopulegon isomerase. IPGI, I can put it here. So, IPGI stands for cis isopulegon isomerase, and IPR stands for trans isopiperidinone reductase. So, R is basically reductase, and this is basically isomerase. Okay. And IPDH is also there, which is basically a dehydrogenase. Okay. Now, next what we see, what is happening in case of carbon. So, carbon, what is this, that the limonene, because limonene moved to endoplasmic reticulum. In case of Mentha spicata, the limonene formed in the plastid moves into the endoplasmic reticulum. So, I will put the limonene here, and this limonene will be converted into trans carveol, TRANSCARVEOL. This trans carveol will come out from the endoplasmic reticulum into the cytosol. So, and this transcarveol will be converted into carbon. So, now I will add the arrow. Well, so you should not be confused. So, therefore, this endoplasmic reticulum I will remove it from here. Instead, I will prefer to put it here inside. So, the enzyme is limonene 6 hydroxylase, which is here L6H, and then there is a CDH, transcarbiol dehydrogenase. So, which converts transcarbiol to carbon. So, this is CDH. So, these two are the major enzyme converting limonene into carbon, and this one particularly you will find in that species. What is that? Which is Mentha spicata. Whereas, the other pathway is operating in the Mentha piperata, the peppermint. So, this is basically in brief, in the most simplified way, the I represented the pathway of menthol and carbon biosynthesis. Okay, one important enzyme I must put it, which is menthol reductase. It should come here. It should be menthol reductase. So, or I can put it, simply write it as MR. MR stands for menthol reductase, and pulegon to menthol formation, this is called PR. That means again pulegon reductase. Well, so this is in brief the pathway, and which operates starting from plastid, involving endoplasmic reticulum and mitochondria, and finally to the cytosol. So, this is basically in brief the pathway. Also, menthol can be converted into methyl acetate. Acetate as well. So, now what we will see? We will see the regulation of the pathway and where we will find the menthol in the plant. So, next slide. So, this is localization of the menthol. So, the place where these menthols are synthesized and accumulate is called trichome. So, I will draw a structure of a trichome to show you here. These are called glandular trichome because this forms a typical gland. I just draw it. This is called the stalk cell, and then above it these are called secretory cells, and then top of it will be the subcuticular oil storage cavity. So, this is This is the oil storage cavity. This is also subcuticular. This is the stalk cell, and this is called the basal cell, and these are called secretory cells. So, the products are synthesized, then from here it secretes here. So, this is basically filled up with essential oil, where menthol is the main component. So, this is basically the structure of one such glandular trichome. There are two types, one is called peltate, another is called capitate. So, if it has a stalk, it is capitate. If the stalk is not there proper, so it is basically peltate. Okay. Now, next is the regulation of menthol biosynthesis. Some problem with the board. Regulation of menthol biosynthesis. So, basically there it can be divided into two periods. One is the two distinct period. One is the first period, so which is basically peaks around 12 days of leaf initiation. That means peaks at about 12 days after leaf initiation. So, what you will find here, in this stage you will find oil glands filling with the essential oil and conversion of precursors to menthol, and also pulegon and menthofuran are also detected, and this will be followed by the second period. So, which is basically one week later. So, in this period what we will see that the transcripts and the enzymes both will be diminished, and then important point is gradual increase of menthol reductase activity MR, resulting in depletion of menthol and concomitant increase in menthol. Okay. So, leaf surface contains glandular trichomes, and when you touch the leaf of this mentha species, basically you are, by touching you are breaking down the glandular trichomes, and if you smell your hand you will get the aroma, because what happens, you are basically breaking down this subcuticular oil storage cavity, which is very delicate, and that is this one. If it is broken down, then that leads to the release of the essential oil. Okay. So, this is brief about the regulation of menthol biosynthesis. I will end this lecture here, and in the next class I will continue this topic to complete the total theme. So, that is menthol story. So, thank you very much.